Hello, everybody. Give me 1 second. Okay. All right. Welcome, everybody. And my name is Japs. And Elia, just wanted to go ahead and if we can go ahead and start.
Yep, let's get going.
All right. Again, welcome, everybody. My name is Japs, and welcome to this session. And I want to start introducing our company, our vision, and team that stands behind the various resources and training programs we will go over today. OmicsLogic is a program for bioinformatics training that was developed in collaboration between Pine Biotech and the Tauber Bioinformatics Research Center at the University of Haifa. The company was started as the result of technology transfer from the center, which was founded by Dr. Alfred Tauber and Dr. Leonid Brodsky. Dr. Alfred Tauber comes from a medical background, has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center at the University of Haifa and is an expert of bioinformatics and biostatistics. And he has numerous publications in mathematics and bioinformatics. And over the course of his career, has developed multiple novel algorithmic approaches to biomedical data analysis. The rest of the team includes experts in bioinformatics, computer science, and training, and some of our team members will be presenting today as well. OmicsLogic Bioinformatics Training offers professional certification based on our award-winning curriculum and supports a research project. We have served over 20,000 students around the world and formed relationships and universities and networks that can now offer easy access to such training in the US, India, Africa, and South America. The goal of OmicsLogic program is to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation for data science. Oops, sorry about that. Enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life and data sciences. We do this by leading each participant through a well-defined learning process. In order to create a clear path of skill development, OmicsLogic employs the Bloom's taxonomy levels of learning. This approach starts with basics, recalling facts and understanding the terminology, ability to remember the material, and with this base, the learning builds to conceptual knowledge, being able to understand concepts and apply information to new situations. Then the goal is to be able to analyze data by drawing connections and evaluate when to use certain methods. This leads to the highest level of learning and the ability to design and create original work. In this program, we will utilize various technical aspects of the OmicsLogic portal designed to make this process flexible. Students can learn anywhere and at any time. Their progress is quantified and activity is tracked using a specially designed point system. After acquiring basic skills, quizzes offer an opportunity to check comprehension. The central focus of this activity is a profile where users acquire levels and badges. Each module will have lessons and assignments that come with an independent certificate of completion. Next, our team will demonstrate the various options so that you are familiar with the process and can make the most of this program. I will pass the presentation now to our CEO of Pine Biotech, Ilya Brodsky.
Thank you, Jabs, and thank you everyone for joining. So I'll share my screen. Okay, so we want to talk today about these summer programs that are coming up. And these include 3 training programs and what we call a research fellowship. And the research fellowship is really a program designed to help students and faculty design and complete independent research projects within the scope of 3 months or this upcoming summer. So the 3 programs that we're going to have are machine learning or biomedical data analysis in R and Python. We'll talk about how to load data, analyze data, visualize data, coding in R and Python. You'll have some exercises, and the objective would be to design and develop a sample project that demonstrates your skill. 2 additional programs are going to be on infectious diseases and chronic diseases. So the infectious diseases will utilize project examples coming from the current pandemic, the previous pandemic or epidemic with Ebola virus, tuberculosis, so different types of pathogens, bacterial and viral. The program focused on chronic diseases will use examples from cancer, Alzheimer's disease, and other conditions, but will be focused on transcriptomic data analysis, including bulk and RNA and single-cell RNA-seq. In general, all of these programs are a part of our basic curriculum, and this curriculum is available for anyone who's here as a faculty member. You can learn more about the different topics that are covered using project examples, and I will demonstrate in a few moments on how these different specialization tracks are created. And they include oncology, infectious diseases, neuroscience, and agriculture or biotechnology. Now, because we deal with a diverse population of students that come from different backgrounds, including engineering and life sciences, we have designed several different approaches to teaching and mastering bioinformatics. And that includes both coding and non-coding bioinformatics paths, where we connect the dots between biology, data, and data science. And the way that we do this is by utilizing several different resources. The first resource that I want to talk about with you today is the OmicsLogic portal. So the OmicsLogic portal is going to be central to what we do. We will utilize it to make sure that everyone has access to practice materials, can really evaluate their learning by completing quizzes, and it's a way for us to offer certification for those that complete a certain number of assignments. Anyone that would like to join the programs that we're going to be offering this summer has to have a profile on this portal. So if you could please go to the link, learn.omicslogic.com. I just posted the link in the chat box also for those of you that would like to follow. On this portal, I will switch over to my my browser. So on this portal, anyone can register for free. And the way to do that is, first of all, to go to the login screen. You can create an account, put in your name, email, and password. I, of course, have a password already and have an account, so I'm going to sign in. Now, once you are signed in, you will see that on top, you will see welcome back and then your email address. What's important to do is to complete your profile. So for those of you that are affiliated with a National Association of Idea Principal Investigators, university lab organization, you have to mention your university, maybe a lab or something like that. For those of you that are from the Louisiana Biomedical Research Network, you can mention LBRN there. So all of this will help us in completing the registrations. We'll talk more about the registration process later, but anyone who will join these programs will have to have an account. That's our way of tracking the activity and making sure that we know who is coming from which university or organization. So this portal will have access to different types of resources, and I'll go over them in detail. What's important is that it will also help you track progress and demonstrate your abilities and skills as you complete different assignments. So there you will see your bio, the point system. We talked already about the different types of subscriptions, and all of this is going to be included and covered by different entities that are going to be different for different students. But basically, this is an important section of the portal where you will find everything that you need to do. And importantly, after completing a certain number of steps, you will be offered a certificate of completion. Now, beyond the profile, there is going to be just a general collection of courses, projects, and examples. And the way to access that is by clicking on the menu on the course tab. You can search for specific topics like cancer or gene expression or something like that. But you also have a number of collections or topics that are covered in many different courses, including project, data science, genomics, BioML, or R. So you can just click on one of these and you will see a collection. There are also dedicated collections at the bottom that are organized by data type and whether you are just getting started or would like to explore different projects that we have. So for anyone that has no background in bioinformatics, and this is not related specifically to any program that we're going to be discussing today, you can simply get started by clicking on the Getting Started button. There you will find just a few resources that are free for everyone forever, that you can go through the basic terminology used in bioinformatics and learn about the applications of bioinformatics. What do you use bioinformatics for? So this is something that you're welcome to share with any connections that you have, students that are starting to learn bioinformatics, really just a resource for anyone to get into this topic. Now, we also have programs. Again, under your profile, you will find several different sections: your activity, courses that we're taking, and then programs. And this is really where we will assign you to the program of your choice. So originally, when you received an email, you were offered a few different options. And when you select one of those programs, you can essentially be added to a program that has a schedule, has a sequence of events. Every event will come with a live session or a recorded session that you will have access to, and assignments that are specifically designed around the topic of that particular session. For each one of these activities, you will get points. And the points are for completing courses, running a pipeline, or running a code block, updating your profile, or participating in a group activity. All of these points ultimately accumulate under your profile, and we have a system for generating those certificates based on the number of points that you receive. For those of you that are interested to pursue an independent project, we also have the option to use those points to then develop an independent project using, again, a system on this portal that I will demonstrate towards the end. Now, in every one of these courses that you find, there will be project examples, and the project examples vary by topic. So here, for example, you can see a collection of projects related to the Bioinformatics for Infectious Disease program, which is focused on different types of pathogen, viral or parasite or bacteria and such. Now, we launched this program for the first time in January for the National Association of Idea Principal Investigators. We offered several different courses, and we're excited to see the progress that some of the prominent members were able to achieve. And the reason we are showing this slide is because you can really see the diverse kind of background of participants in these programs, ranging from people that are maybe research assistants in labs, clinicians, associate professors, students, PhD students, graduate students, and undergraduate students as well. So really, we hope that this resource and the programs that are offered are going to be adapted to the different levels and needs or objectives of everyone in this meeting today. So let me now talk specifically about the summer programs, what each program is going to include, what are the levels, the time commitments, what are the opportunities to complete the program, what will be the result of each program, and things like that. And as I'm going through this information, please feel free to share any questions that you have in the chat. And at the end, I would be happy to address those questions specifically. I already know that some people are wondering about the process to actually confirm your registration. We will talk about that. We know that some people have questions about how many people can participate from which specific universities. So the reason why we will do this at the end is because this is a little bit different for the different groups, for those from the Louisiana Biomedical Research Network, from LSU Veterinary School or other departments at LSU, and also for people in institutions affiliated with the National Association of Idea Principal Investigators. So we will have 4 program choices. You can only select one, but for every one of them, you can also add a basic subscription that will get you introduced to the different topics and the basic terminology. So these include machine learning or biomedical data analysis in R and Python, bioinformatics for infectious diseases, transcriptomics for chronic diseases, and a program called research fellowship. So these 2 programs are intermediate level. Again, the beginner level, if you're a complete beginner, you're welcome to choose getting started with bioinformatics for absolute beginners. After you complete that, you should be ready for any one of these intermediate programs. Transcriptomics for chronic diseases is mentioned here as advanced, and the reason for that is because we will be going through both bulk and single-cell RNA-seq. It will require some background in coding, and it will require an extensive understanding of the biology of transcription, because we'll try to look in depth at the different types of pipelines and analysis methods that can explain what is going on at that transcription level. So that is why this program is going to be advanced. It's going to be a lot more material, and it's going to come with an expectation that you already know a lot of the background. We're not going to go over the basics. In terms of the Bioinformatics Research Fellowship Program, it is mentioned here that it is for all levels, and the reason is because several different scenarios are possible. So the first one might include just doing literature review on a specific data set or a specific collection of articles, research articles. You can also perform a simple research project where you just take a single data set and you kind of use standard methods to analyze that data set and present your results. Or you can have an advanced project, and that advanced project might require multiple types of data or several different large data sets and maybe complex methods that will require some time to process and complete, and then a poster presentation or potentially even a draft for a publication. So we've seen a number of students and faculty that have completed in the course of 3 months a full project and have resulted with a publication, even though that obviously cannot be guaranteed. It takes a lot of effort and a long time to do that. So how are we going to support all of these different types of programs, and what are the resources that you will have access to once these programs start? By the way, I forgot to mention that for those of you that are from LSU and from LBRN, we do have a specific registration process. For the Louisiana Biomedical Research Network, it's a different process. For LSU students, it is different. And for NAIPI members, it is also different. So before we go on, Farhana, would you like to say a few words about the registration process for LBRN and LSU?
Yes. Hi, everyone. My name is Farhana Musarat. I'm the program coordinator for the summer virtual training program. And let me share my screen. Although most of the things are already explained very well by Ilya and Zaps. Okay, so yeah, so I am the program coordinator for this course, and I'm also a postdoctoral researcher at LSU School of Veterinary Medicine. So as it's already mentioned that this course is actually open to all, so everybody can take these courses in summer. However, we offer a free registration for at a limited number of people for LBRN members and LSU School of Veterinary Medicine members and NAIPI. However, if you are part of the LSU, like LSU main campuses, if you are part of the LSU main campuses, like if you're faculty or students, you can also take these courses, but you have to pay for these courses and it has to be through your department. So if you are interested, please email me and we can arrange, check like whether it's possible or not. And for other people from LSU, SVM, NAIPI or LBRN, it is gonna be free. And the registration process after you all register through this link, as you already saw in the flyer, when you open an account and register through this link, we will have your email address and names and informations, and we will contact you, and detailed procedure will be explained, like how to register for the for specific courses that you are interested. So I guess this is it. And if there's any other questions regarding the registration process or the courses, you can email me. My email address is already in the flyer, and I will also write in the chat section here.
All right, thank you, Farhana. And yes, I already put your email address in the chat box there.
Okay, thank you.
Okay, so again, we can connect further if you have any questions about registration. But now let's talk a little bit about the resources that you will have access to and how exactly this is actually going to work. So first of all, the programs are different from courses because they include interactive sessions. They include assignments that are linked to the specific topics of the program, and you will have access to big data processing capabilities. So if you actually have a data set that you would like to analyze, you will have the opportunity to do so, and we'll go through a number of different resources in different programs that are more appropriately designed to address those kinds of data sets or data types and those kinds of projects. So first of all, I want to kind of talk a little bit about the fact that this is intended to make bioinformatics more accessible to anyone. And so anyone that you think might not be a good fit for the program, they don't really know anything or something like that, we do recommend trying out to sign up. And there are two specific courses. There's a course for Introduction to Bioinformatics and a course called Bytes and Molecules. And both of these courses are designed specifically for those that either have not touched bioinformatics in a long time or would like to just get started. And that's free and accessible for anyone around the world. So you're welcome to just go ahead and do that. And specifically, this is designed because there is a need in bioinformatics training and skills for anyone who's involved in biomedical research. Now, for those of you that are going to be joining the actual programs, the programs are going to include access to the training materials. You will have access to data analysis tools, curated project data sets, and interactive sessions with a variety of faculty members. And I just want to mention that you will also have access to a library of pre-recorded lectures that are contributed by different members of the programs that are represented here as well. So in the past, we've had a number of guest lectures from the Louisiana Biomedical Research Network, from companies, from research institutes, from the administration of the different programs, and really getting access to all of these in the context of a specific program is going to be kind of opening up further opportunities for you to learn and explore specific opportunities to complete a program or even continue your education beyond what we can offer online. Now, an important resource that we are going to be using throughout the program is a user-friendly interface that allows us to build large data processing pipelines interactively without any coding, where you will have access to 5 terabytes of storage, cloud containers that you can deploy to perform complex analyses, and technical support. So if something doesn't work, you have someone to talk to. This platform is embedded in the educational resources that we will go through because it combines the logic of analysis by using color-coded interfaces, by offering an explanation about each method with links where you can explore them, with a variety of output and input formats that you can kind of intuitively understand. And we will go through these examples so that you can appreciate, first of all, the diverse nature of data using different types of sequencing techniques and the diverse nature of projects. And obviously, every project requires some preparation, some processing, and to skip that part, just to conceptually understand and appreciate it, we have this resource. After the program is complete or during the program itself, you will be able to utilize the platform itself to perform your analysis. So all of the different sections that we will go through will actually have working resources that you can use. Now, in addition to that, we're going to go from context to logic, practice, and then to independence. And as a result, many of the analyses that you will perform, you will be able to take away together with you as an interactive notebook. That notebook will contain code, will contain access to the data and visual output. So you will actually be able to hopefully go from a very basic introduction to what bioinformatics is all about to practically using bioinformatics without any technical necessary steps, and then eventually being able to interrogate that data in a way that you can both understand and share. So let's first talk about the biomedical data analysis using R and Python. So here, obviously, one of the challenges is to leverage both of these languages to understand how they can be used to perform the same thing. And typically when we say bioinformatics using R and Python, we talk about producing plots, performing statistical analysis, and an introduction to machine learning. So the way that this was going to work on the portal, I'm just going to switch really quickly, is by doing the following. So again, if you have already created an account on the portal, we can take a look at one example. So if you type in introduction to, you will see that there's going to be an introduction to Python, an introduction to R. So I'm just going to demonstrate very quickly what that looks like. In each one of the courses, you will have some lessons that are available for free, and those are the ones without the star. And then the ones with a star are only accessible through a subscription. Now, that subscription is what essentially will come with the program, so you will have access to all of the basic as well as the premium materials. Here we can look at one example that should be accessible to anyone, where there's an introduction to plotting. And you will see that each section will include an overview of the syntax so that you can understand how code is written, with the ability to actually understand the results. So you can see that these results are representative of what you should get as a result of running code. And then you will also be able to practice this code right in this section as well. So for example, just simply running this code is impossible. I have to understand what is being done. And once I do this, I will be able to run and complete the assignment. And as you progress, it will become more and more complete, more and more complex. And so once you have completed all of these assignments and you're able to understand how to load data, visualize data, and do exploratory analysis and so forth. So this program in parallel will offer examples in R and Python. You can choose either one. You don't have to just try to accomplish both of them. At the end, as these courses become more complex, you will also have access to interactive notebooks where you can run a full code and submit that to somebody who can review and guide you if you have any questions or any challenges, actually becoming independent with those methods of analysis. The reason we are focused on R and Python are for two reasons. The first is that this is the most demanded skills in the industry, but also because a lot of publications leverage visual outputs from packages that are commonly used in both of these languages. So it will start with an introduction to both R and Python. And then you will also get an introduction to data science in R and introduction to data science in Python, which covers a variety of topics like data wrangling, machine learning and statistics, unsupervised and supervised types of analysis, and other types of methods that are listed here in detail. Now, this starts from very basic things like being able to load data, understand matrices, being able to call on specific rows and columns. Then it will go through more complex analyses. And the way that this is done is structured in the same way that was already explained, from understanding the problem to then being able to learn the code, modify outputs, settings, and then finally apply to a specific data set. Now, in all of these methods, you will find a coding challenge, an interactive notebook, and assignments. And these assignments, again, could be reviewed by the guide of the program so that you actually can overcome any technical challenges that you will face. Now, importantly, many of the principles of exploratory analysis are specific to a different type of data. And we will try to incorporate examples that show how data analysis using traditional methods for exploratory analysis and more advanced and more adopted more recently have been changing as a result of the changing nature of the type of data and the quantity of data that is being produced. We will talk about how to evaluate different methods, and we will also think about when to apply different types of methods to what kind of challenge, knowing that we can oftentimes be looking at different stages of the analysis that actually combine all of these methods together in a single workflow. Now, at the end of kind of an overview of different methodologies and use cases, you will be introduced to projects. And those projects will include examples from diagnostics and cancer biology to single cell and tissue composition, and projects like microbiome and infection. So again, if you have any questions about this first program, this is an intermediate level program, so you need to know a little bit. Definitely don't be intimidated by the program. Anyone will be able to successfully complete all of the assignments. Always, if you have any trouble, someone from our team will be able to assist you with that. Now, the second program that I want to talk about is Transcriptomics for chronic diseases. The program is designed to address commonly used methods for data preparation, which is a pipeline to process FASTQ files and turn them into a table of expression for gene or isoform expression, statistical analysis of differences between groups of samples, like t-test, or eventually adapted into packages like DESeq, DESeq2, EDGE-R, and others, where you will be able to perform the analysis, explain it in a biological way, and also produce the different types of plots that are commonly used in publications. Now, we will also focus on single cell here as well, and that is why we call this program more advanced program, because it will try to kind of cover everything that is today used for transcriptomic data analysis. And single cell comes with a whole range of complications that are different from bulk RNA-seq, and learning everything in a single program will require a little bit more effort. Now, the reason why we study transcriptomics is, of course, because it's a good proxy for translation for the proteins that are being produced. Specifically in the program, we will talk about next generation sequencing as opposed to some of the other types of methods of quantification of RNA. And so we will skip microarray and kind of Western blots and things like that, and we'll focus specifically on next generation sequencing. And we will see examples of projects that apply these techniques to a number of different directions, including biomedical research topics like diagnostics and therapeutics in the era of precision medicine, especially focused on examples from cancer biology and biomedical studies in that domain. The topics will give you both an overview of the different resources for transcriptomic data analysis, and all the way to some hands-on examples that will be curated and designed to illustrate a specific understanding or a specific topic. Now, for those of you that are not interested in necessarily becoming proficient at coding, It is also going to be enough to just understand the logic of the analysis, and we will utilize the T-Bioinfo platform to make sure that the logic is clear, because it provides a number of resources that will also guide you as you try to apply these techniques to a data set of your own. You will also have access to interactive plots that you can explore, and downstream interpretation, including pathway analysis, gene set enrichment analysis, gene ontologies, and others, as well as interactive outputs for exploring the types of mapping techniques that we're going to utilize, and also exploratory analysis methods. In the era of bulk RNA-seq becoming more accessible, the prominence of machine learning has increased. And so a lot of people leverage big data sets like the Cancer Genome Atlas and others to explore associations that are more complex than what typically could be done with differential gene expression or linear regression. And so we will look at a number of machine learning methods specifically applicable to continuous data used in RNA-seq. And there we will look at decision trees, different types of discriminant analysis, and classification methods like random forest. Now, when we transition to single-cell transcriptomics, we will discuss the common methods of data preparation, as well as common methods used for analysis of single-cell RNA-seq, including dimensionality reduction, UMAP, t-SNE, and others, and pseudo-time trajectories, so that we understand both technically how those should be performed, but also in terms of biological interpretability, how do we actually use some of these methods to get the types of plots and results that typically are presented in research articles. As I mentioned, all of the programs will be supplemented by project examples. These include analysis of different types of projects, and these projects are going to range from cancer to infectious diseases. And that brings me to our next program, Bioinformatics for Infectious Diseases, which is going to cover some overlap of topics. It will include transcriptomic data as well, but it will focus specifically on the way bioinformatics is used to study the actual pathogen as well as host pathogen interaction. And so that is why we'll talk about analysis, first of all, of genomic data, and also analysis of the host pathogen interaction in genomic and transcriptomic data. So we'll talk about topics like sequence variation and alignment, evolutionary and phylogenetic analysis, functionality and protein structure. How do we map variants onto the protein structure, excuse me? How do we analyze 3D structures of proteins? And then transcriptomic analysis of host response. So to do that, we kind of first have to start maybe by talking about what is the difference between chronic diseases and infectious diseases. And the main difference, of course, is that infectious diseases are caused directly by a pathogen and they spread. And the chronic diseases are caused by environmental and lifestyle choices, and they typically do not spread. And so because of that, we will take examples that will help us understand how these pathogens are different and how do they actually cause what we call disease, and what are the specific functions that we would like to understand. Now, the study of pathogens, of course, is one of the first interests in microbiology, but also it is a first example of bioinformatics leverage to actually, in almost real time, analyze and understand the evolution of the pathogen as it spreads in a population. So the first example of a very successful utilization of bioinformatics in the context of an epidemic was in the 2014-2016 Ebola outbreak, even though bioinformatics obviously has been used before. But during that time, in a very short period of time, researchers were able to track the evolution as well as design interventions that are both population level and personalized vaccines and antivirals that became successful and are now being used to kind of address these outbreaks. So we're going to use that example as one of the starting examples of how we can use bioinformatics effectively to do that. But beyond that, we will obviously use the current pandemic as one of the examples that is still ongoing. We'll talk about how in this pandemic specifically, different types of methods have been used to understand the interaction between the host and the pathogen. How does the pathogen enter the cell? Where can we find the specific mutations responsible for adaptation? How can we identify protein targets? How can we think about the intervention and think about the relationships between the different strains of the pathogens? Now, to do that, we will try to understand different types of sequencing techniques that have been utilized in the different examples that I just mentioned, and obviously the specific needs for the analysis that we will discuss. And we will talk about how these different types of data are used, where to find them, where can we find collections of data about these different types of diseases, and what are some both open source and kind of available online tools that people use, and what are some of the methods that are more custom, and how to ask questions from these types of data sets. So bioinformatics for infectious diseases will include kind of a range of topics and a range of different types of data so that you can access all of these different tools. The final program that I want to talk about is called the Research Fellowship. The Research Fellowship is a different type of a program. It does not have a set curriculum because really the objective of this program is to provide training in the context of developing an independent research project. So to develop an independent research project, your objective is to actually design a research poster or a project that you could present to others. And so with that objective in mind, what we will do is we will have access to big data, mentorship, and training, and that will be divided up into several different topics or domains. And so these domains include precision medicine, like cancer, neuroscience, and others, population health, like infectious diseases, biotechnology and agriculture, or even space and astrobiology. So the way that you can design and complete a project during the short period of time is by following pre-designed steps on the process of identifying a question, finding a data set that you could ask that question from, knowing how to analyze the data to produce the results that you need, and then presentation, or how do you summarize your findings in such a way that people understand how you arrived at those conclusions and what are the actual research findings that you are presenting. So it all starts with literature review. Many of you probably already know how to do literature review, but in the context of bioinformatics, literature review involves several additional steps. First of all, we do want to find existing resources that could be categorized in databases, maybe pipelines, or examples of methods and how they were applied. In many cases, you are looking for gaps, controversy, and debate. And so a lot of people in the domain of bioinformatics compare and contrast different pipelines, maybe evaluate the comprehensiveness of a resource or a database. And in many cases, you can then identify a question or a hypothesis specifically structured around the area of your interest or your expertise. And then our program will involve several different steps, including an introduction, which will give you an overview of different types of methods and tools that you can use, evaluation, so that you can propose a project and get feedback on that proposal, and then practice, so that whenever you are ready to perform the analysis, you're actually understanding and you're equipped to be able to do that independently. So the outcome of that is to complete a project and submit it on our portal. And to get there, you will have access to mentorship, someone to review your proposal, technical support from our team, and a community of like-minded students that you can interact with and potentially design even group projects together with them. So on the portal, you will be able to utilize our system to guide you through all of the steps that are typically included in preparing a poster presentation. And here I have a brief recording of how this is possible to do on the portal itself. So the first step would be to complete your profile. Then you would have to add your bio and connect to your OrgID or your social media. And after you do that, you will have to earn 10,000 points by practicing different types of analyses that we describe in programs and courses, and complete a course that speaks about the way that you can utilize bioinformatics to do a research project. Now, after that, you can start your own project. And the project will have several different sections, including an overview. Reproducible projects include a reference data set that is curated and prepared. So we will go through the process of submitting that data set on Dataverse and explaining the metadata. We will then talk about all the standard sections like abstract, introduction, methods, results, discussion, and conclusion, including a list of references and acknowledgements at the end. Now, as a result of completing such a project, you will be able to add that project to your resume or present it at a poster conference. And so here we have several examples of people that have submitted either as a preprint, others have published in the undergraduate journals, and others have even used some of the results that they were working on in their ongoing research studies. Some just use that example to train and acquire skills as well. But what's important is that at the end of the summer, in August, we will have our virtual Omics Research Symposium. This symposium is a biannual event that we organize together with our partners, where we invite experts from bioinformatics research in industry and academia, as well as experts in bioinformatics education, to discuss pressing topics and advances in bioinformatics, but also we provide a platform for students, faculty, and researchers to present those posters and get feedback, specific feedback on their projects and how to take those projects further. So it's really an opportunity to find mentors and communities that you can join and learn about how to implement this kind of training wherever you are. So again, with that, I am happy to take any questions that you might have. I will briefly mention the process of, again, registration. So for any one of you that is coming from NAIPI, from the National Association of Idea Principal Investigators, one of your questions might be, well, how do I know that I am from NAIPI, right? How do I know that I'm affiliated with the NAIPI institution? The way to know that is to go to naipi.org, which is the website that lists out all of the institutions and all of the states that participate in this program. So if you just scroll down a little bit, you will see this map. On the map, if you click on any one of these, you will find a description. And if you scroll down beyond that, you will also see all of the active principal investigators. So to participate in the program from NAIPI, you have to be a member of the institution where the PI is at, right? So if your lab PI is a member of NAIPI, that's great. If in your institution there is a PI, that is enough, right? So really, this is just the institution. So for example, in Louisiana, people from LSU, LSU HSC, etc. Now, we only have a limited number of NAIPI licenses that are going to be covered 100%, and that's 50. And then we have an additional 100 that are going to be offered at 75% off. So that's still a significant discount. And so I think it would be useful if you also don't know whether your institution is a part of NAIPI, you can simply search for that institution. Yeah, so for example, oh, this is Rhode Island. I think the way you search is by going, 1 second. I think you have to go to the state first. So actually, I'm not so familiar with names of universities in every state, but in any case, you should be able to find, yeah, and you can also go here by region, and you can see the different actual projects that are funded by the IDEA program, but by the NIH IDEA program, and NAIPI just brings together all of the PIs from those funded projects. So again, this is for NAIPI. How do you register if you are a part of NAIPI? So all you need to do is you need to create the account on learn.omicslogic.com. Once you have created an account, when you are making that account, it will ask you what institution you're from, and you will have to add into your bio that you're a part of NAIPI, LBRN, or LSU, or any one of these institutions. So please do that and let us know. So we have here Japs, who is going to be able to assist you if you are from NAIPI or from any other university for that matter. Farhana, again, can be helpful for those that are from LSU, LSU SVM, or from LBRN. And we also have a specific process for those of you that are from LBRN. So from LBRN, you do not directly go to us. You actually go to this page, and John here from LBRN has already shared this link. So the link, I'm going to post it again, lists out all of the different programs that we just discussed, and then has here at the bottom of the page the registration link. So if you are from LBRN, you click there and you just process from there. And what will happen is once you submit that application, you will be reviewed. And if it is granted, then you will be informed about that decision through LBRN itself, not through Pine. So you will have an email from someone within LBRN that will notify you about you being accepted. And I believe that the last day of registration is May 27th, even though that might be extended into the first week of June. John and Farhana, correct me if I'm wrong here. Making sure. Yeah, John, I don't know if you want to.
I was trying to unmute and you.
Yeah.
Yes, that's correct. You are correct about that. I've been getting a lot of non-LBRN participants trying to apply on our form, so I'm having to decline those, unfortunately, and I'm relaying that information. But please, people, when you look at the form, it does very clearly state which institutions you can apply from, and they have to be from Louisiana. Thank you.
Perfect. So thank you. Now, for those of you that are not from any of these institutions, so if you are not from LBRN, you are not from LSU, you are not from a NAIPI associated institution, you can still apply for these programs. The way you do this is you let us know. So the best way would be to email hello@pine.bio, and that is the email address for Japs. She will be able to identify which program is the best for you, have a conversation with you if needed, and then recommend the best path. So there are several different pricing points from beginners to advanced with different kind of levels of access that you can get for different links. For those of you that are interested in a program, not just over the summer, yes, those are available as well. Again, you can go simply to the program tab. And on the program tab, you will see different types of programs and different dates and things like that, and you can explore those. You are also welcome to email us about them if you have a specific program in mind that you're interested in a specific time frame, in a specific location. We run these at different time zones with different types of trainers, and so we should be able to help you with that as well. So hopefully, Anna, that answers your question. So as you saw today, we cover many different topics in bioinformatics. I want to make a final note to those of you that are faculty. If you are a faculty at any of these institutions and you would like to include any of these resources in your training, in your coursework throughout the fall and into the year, you're welcome to utilize these resources as well. We have guides and all kinds of resources to support that. You're welcome to reach out to me directly if you are a faculty member or you are interested in that, and we can discuss the options for that as well. If you would like to introduce your own material and you would like to share that material with the National Association of Idea Principal Investigators. I also want to mention is that one of the objectives of the National Association of Idea Principal Investigators is to coordinate sharing of resources related to bioinformatics. And so if you go on the NAIPI website under resources, and there's a tab here, training resources, our OmicsLogic programs are listed here, as well as some of the other resources that are shared by the members of this community. And you can even submit your own resource here at the bottom as well. So if you are teaching bioinformatics, or you have a summer program, or you have a program that others can leverage for free or paid, this is a place to submit that program and make sure that the community knows about it. So please do that. And if you have any questions about that, you can simply go to this page. All right, so thank you everyone again for joining today. Excited to get this program off the ground soon. We only have a few days until the official registration date is going to be closing. And then after that, we will have some room to kind of accept those that are still not sure. But please do share this with your network. We will post this video on YouTube as well, so you can share that. You will receive an email from us with that recording. And thank you again for joining today. We look forward to interacting with you as the programs begin.